A regulation of gene expression. Gene expression is the synthesis of a gene product by a gene. The gene product is a protein or an RNA. The protein is produced by protein coding gene. The protein coding gene produces protein through two steps, namely transcription and translation. In transcription, the gene produces mRNA. In translation, mRNA produces protein. The non-protein coding gene produces RNA, rRNA, tRNA or snRNA, that is small nuclear RNA, by transcription. Protein produces phenotypes of organisms such as the morphological character and metabolic changes. Regulation of gene expression is the switching on or off of genes, and when, how long, and to what extent the genes are to be expressed. Gene regulation is the modulation of gene expression. Gene regulation ensures that the appropriate genes are expressed at the proper time. It helps the organism to increase or decrease the required gene product. Need for gene regulation. Control of the cell structure. Second one, control for cell function. Third one, enzyme reaction. Fourth one, metabolic pathways. Fifth one, cellular differentiation. Sixth one, creation of different cell types. Seventh one, morphogenesis. Eighth one, embryo development, adaptation to environment, and substrate for evolution. And the reason for regulation of gene expression. The cell develops from an egg by repeated mitosis. Hence, each cell contains the full set of genes as in the egg. If all the genes are producing proteins, the cell will be flooded with proteins. It is wastage of energy. Hence, all the genes in any cell will not function. The genes not required are switched off. Each cell has a specific function. The gene related to that function along is switch on. The genes not required are switch off, and the genes related to that function along is switch on. For example, in pancreatic cell, the insulin producing genes are switch on. Some genes are essential for the survival of cells. These genes are called constitutive genes or housekeeping genes. They are kept switched on permanently. And the genes which are switched on when their products are required are called facultative gene. And next one, regulation of gene expression in prokaryotes. Regulation of gene expression is well studied in the bacterium, that is Escherichia coli. A prokaryote, the gene producing regulated enzymes are called facultative genes. They are switch off when they are not needed and switch on when they are needed. In Escherichia coli, certain enzymes are continuously produced regardless of their needs. These enzymes are called constitutive enzymes. They are permanently switched on. These enzymes are produced by constitutive genes or housekeeping genes. They require no gene regulation. For example, enzymes of glycolysis. Certain other enzymes are synthesized only when they are needed. Such enzymes are called regulated enzymes. They require gene regulation. Example enzymes of lactose metabolism. Bacteria are simple unicellular organisms. The chromosomes lies in the cytoplasm. There is no nuclear membrane, so the regulation of gene expression is also simple. In Escherichia coli, regulation of gene expression occurs at two levels. They are regulation at transcription and second one regulation at translation.